Okay, good morning. Good morning, everyone. My name is James Polli from the University of Maryland, and welcome back to day 2 of the MMF workshop. And to give some opening remarks this morning is Huang from the FDA Office of Clinical Pharmacology, where she's Deputy Director. I think all of you know who she is. She has many interests in over quite some time in clinical pharmacology. My little story about Shiew is that she gave me a baseball cap. She gave me a baseball cap that says FDA Office of Clinical Pharmacology. And I don't know if Joga has one. Okay, Joga has one. I think Tim from Pfizer, he was asking about it. He wanted to know if I was going to sell it on eBay, and I said no. So I still have it, and I do wear it because I actually wear a lot of baseball caps. Shiew.
Thank you, Jim. Good morning and good afternoon, depending on where you are. So it's day 2. Welcome, everyone, to this workshop on consideration and potential regulatory applications for Model Master File. Many of you know that the FDA has had a half-a-day workshop to introduce the Cedar Quantitative Medicine Center of Excellence. It involves the development and application of exposure-based biological and quantitative modeling and simulation using non-clinical, clinical, and real-world data in order to inform drug development, regulatory decision-making, and patient care. So we can champion, advance, and integrate quantitative medicine for maximizing patient and society benefits. And I have listed here the link and the email for further communication. Okay, so let me see. I do this one?
This one.
So what did you do?
I just double click.
Double click?
Yes.
Okay, I'll double click. So at the workshop, Raj Madhabushi has indicated the scope of the center, and he used examples to show the pharmacostatistical models, mechanistic modeling, machine learning and artificial intelligence, biomarker endpoint development, clinical trial simulation, and in silico predictions. And further, Zhao discussed the, thank you. I will not touch it. The evolution of the quantitative tools, starting with PK, biopharm, IVIVC, POP-PK, I'm looking for my red arrow. ER disease modeling, and the expanded scope for quantitative medicine, including PBPK, artificial intelligence, machine learning, QSP, digital biomarkers, real-world evidence, real-world data. Some of them you will hear today. Okay, God, it worked. So yesterday we heard from drug developers, software developers, from academia, regulatory agencies, the FDA, the EMA, MHRA, on a model master files. And I selected 3 slides just to review. So Rob Lineberger discussed the advantages of using model master file. They're reusable, scalable, and support the ecosystem for model development. And the FDA assessment questions are consistent across applicants, and also it can support innovative approaches. So from one of the drug developers discussed opportunities and challenges. So for example, the advantages and the opportunities are incentives and reduced product review timelines, the publicly available file that can be used by multiple applicants. But on the other hand, there are many challenges, such as some models may not work for other applications, and whether the sponsors are willing to share publicly their models. Further, from one of the drug software developers also discussed the practical considerations. So very detailed questions. And I have to thank those that let me use their slides, Caliparo and now Dr. Patel. So what would be considered sufficient justification for modeling platforms? And would peer-reviewed publication be considered sufficient to support VMV a modeling platform? So where would such data be submitted for MMF review? And critically, how would it be reviewed, stored, updated, and distributed? So this is about platform. For drug and drug products, Dr. Patel has listed 5 or 6 considerations. One of them is when there are multiple values of a given parameters, they're available for a particular drug. In the literature, which value should we use in MMF? And who will be responsible for deciding the acceptable value? So from yesterday, I think we have in-depth discussion on the utility and challenges, and also started with the defining the framework and using the oral dosage form and long-acting injectables as examples. The in-person discussion, I think it's very helpful. So I'm looking forward to today, additional thorough discussions for another fruitful outcome for today. Thank you.
Thank you so much, Dr. Huang. Good morning, everyone. I'll be very quick in providing the overview of day 2 of the workshop. I would like to welcome you back to the second day of the workshop, the Model Master File workshop on considerations and potential regulatory applications. I'm sure that you will agree with me that yesterday was a busy but also very productive day. We discussed the MMF workshop. We were given the opportunity to express concerns, challenges, but I feel that we have also identified opportunities regarding the Model Master File framework. It is my impression that the content of day 1 allowed for certain attendees to better understand the utility of the Model Master File and recognize its promising nature. Yesterday, we delved into discussions on potential applications of the Model Master File for oral dosage forms and long-acting injectables. Today, we have planned 2 sessions, as you can see on the slide here, 3, but I'm going to focus on the first 2 first. The first one will take a deep dive on how we can improve regulatory acceptance for dynamic tools, in silico tools for innovator drugs. And the second session of the day, which is my personal favorite, we'll continue the discussion on Model Master File applications for locally acting drug products in the generic and new drug space. During the second session, we will hear the regulatory perspective from FDA as well as EMA, as well as industry representatives will share their thoughts on the potential of the Model Master File framework, specifically for orally inhaled drug products and drug products that are applied on the skin. During session 3, Dr. Amin Rustami will open the small group discussion session, which will be the last session of the day. And the in-person attendees will spend 2 more hours discussing the model master file framework, following the same pattern and arrangements as yesterday. Finally, Dr. Liang Zhao will end the workshop providing his closing remarks. So let's kick off day 2 of the workshop with session 1, Pathways for Regulatory Acceptance of Dynamic Tools in the New Drug Space. And I would like to welcome on stage Dr. Jiang Liu. Thank you.
Hi, good morning, everyone. My name is Jiang Liu. I am Associate Director in the Division of Pharmacometrics, FDA. So in this section, we will discuss the pathway for regulatory acceptance of dynamic tools in new drug development. So we will have 3 presentations. So first is from Dr. Hao Zhu, our Division Director, also in the Division of Pharmacometrics, FDA. He will briefly introduce the fit-for-purpose program for new drug development. And the second presentation is from our PBPK leader, Dr. Yuching, and she will introduce our current thinking on practice in model PBPK model evaluation and assessment for models reusability in new drug development. And the third one is from Dr. Joga Gobburu, who is the well-known professor in the University of Maryland, Baltimore. And he will share his thoughts on the potential of repeated usage of PBPK model to support the BE assessment in new drug development. So following the presentation, we will have a 30 minutes panel discussion. We will also have 3 additional well-known experts in this field, Dr. C.J. Masad, and she is the Vice President of the Scientific Research from Pfizer, and Dr. Martin Klein, and he's the Senior Statistician from FDA, and Dr. Liang Zhao as well. So welcome, Dr. Zhu.
How do I get this? Well, thank you, Jiang, for this introduction. I really appreciate the organizers for providing us the opportunity. So be happy to give a brief introduction for this fit-for-purpose program. It is a program that we actually applied it to accept what we call dynamic tools, essentially reusable models or model framework in the new drug development space. Of course, I have a disclaimer. So what I'm presenting here today are just my personal views. It should not be understood as the official position from the US FDA. It doesn't work. How come? Okay. Oh, where it goes? Ah, they're going backwards now. Sorry. How do I get back to? Left click.
Yeah.
Oh.
See. Oh, there's a clicker.
Oh yeah, this is much easier. Okay, thank you.
Let's go with that.
Okay. It doesn't go. It doesn't go either. Okay. Oh no. Oh, moves too fast. Okay, here. Yeah. Okay. Thank you. So sorry about this technical issues we're having here. So again, my presentation includes the following section. I'm going to start by giving the general background about the modeling tools we have seen in new drug space and what are the features we have seen so far. And then I'm going to move on to talk about this program, fit-for-purpose program. Essentially, like I said earlier, this is a program that is designed to accept the dynamic tools or reusable models with some case examples. Then I'm going to conclude with some take-home messages. And then I'm going to also talk briefly about some of our general thinking for how this model master file can be potentially applied in new drug space. So with that, I'm going to start by looking at the modeling and simulation tools we have seen for new drug development. There are many features if you look at what we have actually tried to apply modeling simulation there. I would say the first important feature is that we have a lot of diverse tools that can be used to support new drug development. Essentially, this is what we call model-informed drug development. So if you really look at the definition, it really refers to the development and the application of exposure-based biological and pharmacological models derived from preclinical and clinical data to address drug development or regulatory issues. So if you open the toolboxes, you can see there are many modeling tools available already. Of course, we can use what we call population pharmacokinetics model to understand the concentration and time course, and also try to understand the variability comes along with this, within what we see in the PK space from the general population, and understand the sources of variability. And of course, we can always try to link the concentration changes to the lab value changes and subsequently to the clinical outcome changes. That's what we call exposure-response relationship. And not to mention, in the recent decades, we try to use mathematical equations to describe the general disease progression and understand how patient features may change the underlying disease progression. That's what we call disease models. And of course, we also try to understand whether the patient behavior features, such as premature discontinuation, how does that really affect the clinical trial readouts? And we can use all the tools in isolation or in combination to drive to address different issues we encountered in drug development. And in recent years, we have seen some of the new trends. We try to use artificial intelligence, machine learning models, or we try to get in more mechanistics. We try to understand, what is the current understanding for the underlying disease and pharmacology, and putting them into mathematical equations, which is quantitative systems pharmacology models. And all these models actually try to help us to modernize new drug development and enhance patient care. And from agency side, we try to use those tools to reshape the policy development. So I think that the next feature is that there are many questions we try to address by using modeling simulation tools. I just try to give you some examples here. On the clinical trial design space, we know that modeling simulation tools are broadly used to address, for example, the dose selection, dose optimization. We're trying to understand what is the appropriate dose, dosing frequency, dosage adjustment. Do we need a loading dose or maintenance dose? I mean, all sorts of questions we can use modeling simulation to help us. And well, of course, we can use trial simulation to determine appropriate sample size. And we can optimize the sampling schedule to make sure we can get the most informative points for selecting, to collecting our samples. And of course, you can use modeling simulation tools to fine-tune our inclusion and exclusion criteria so that we can have most appropriate patients in the trials. And also we can use that information to conduct a so-called stratified randomization or enrichment strategy. Of course, we can use modeling simulation tools to help us to select the biomarkers, endpoints, things like that. From regulatory side, we've been using the modeling simulation tools in many ways. For example, we can use modeling simulation tools to identify what we call substantial or confirmative evidence in order to support a product approval. Another token will also try to use modeling simulation tool to justify that we can extrapolate the efficacy findings from one population into another population. And we can try to use those tools to find what is the appropriate dose so that we can label in the package insert so that we can inform the health providers how to use the drug properly. And also we can use these tools to address risk and benefit issues, and also identify potentially high-risk patients or identify potentially patients who will be good responders. There are many questions we're trying to use modeling simulation to address in today's new drug development paradigm. Now I'm going to move on by using drug dose selection as an example to demonstrate how we actually see the model building and decision-making in this new drug development space. Essentially, you can see this, this black line that represents drug development space. So starting from the first-in-human study, that's where we probably have the largest uncertainty about what is going to be appropriate dose for this patient population to begin with. And what we can do here is we can use modeling simulation to integrate all the pieces of information we obtained through preclinical findings, some of clinical experience from drugs with a similar mechanism of actions, and also we can use some of animal study findings, and we can put all the pieces of information together to come up with what we think is appropriate or potentially appropriate dose range to begin the first human study. So as the program gets progress, with time goes by, then we can collect a lot of clinical data, and each time we can actually fine-tune the model and use the model to derive what we think is more, the appropriate dosing for the next phase of clinical trial. So essentially what you see is that as the clinical data accumulated over time, our uncertainty in the dose selection gets less and less. But what the model does is actually, in this case, is really the model building process is really an evolving process where we always try to fine-tune the model and using the model to drive the next phase of decision-making, so it becomes an iterative process. So, but do we have reusable model? Yes, we do in the new drug space. And one good example is the disease model, which we think is an appropriate tool to address trial design features across multiple programs. And I'm just giving one example here is a disease model for childhood onset dystrophinopathy, which is, it's essentially a very rare disease, but it's it could be deadly as the patient grows older. And what we've been trying to do here in those paradigm, we try to collect all the clinical trial data across multiple clinical programs, combine them together. And also we try to get some natural history findings together and use all available data to understand what the disease progression looks like by using appropriate mathematical equations, and also identify what would be a important prognostic factor for those patient populations, and also try to understand what discontinuation may affect the outcome of changes. But the question we're probably generally facing is that are we going to evaluate the reusable model in the same way as a single-use model? So the answer is it really depends. So now I'm going to really talk about this, what we call model risk assessment framework. I know we have seen this framework yesterday, but I think this is going to be very important for us to understand what type of model evaluation activities and standards needed after giving this exercise. So there are actually 5 key elements we wanted to go through in order to determine what is important for us to understand what level of model evaluation activities were needed. So the first level we wanted to address is, what is the question of interest? Essentially, we're trying to understand what type of a question we wanted to address by using either clinical trials or modeling simulation tools. The context of use specifically talk about how this model is going to play a role to address those questions of interest. And there are 2 other variables which are also very important. One is what we call model influence. Essentially, you can understand is the contribution from model to the totality of evidence. It could be understood as a ratio. And decision consequence says actually based on the totality of evidence we have, there is always a chance for us to make a wrong decision, which can generate a potential risk to patients. The risk could be understood as a lack of efficacy or some of the safety risks. So then you combine the decision consequence with model influence, you get a model risk. So the risk is going to determine what would be appropriate model evaluation activities and standards. So what I'm going to try to see here is that for a specific program, let's say I have a disease model, I'm just trying to apply this disease model to support a specific program. It's probably easier to determine what is going to be the model risk, because I know what the model influence and decision consequences is exactly what it is, given the information we have given totality of evidence we have and given the information we have for this program. But for a reusable model, it's going to be a little bit more challenging because we don't know what is the program generally look like. So what we can do usually is try to think about more conservative criteria, because we might consider that, for example, the model actually plays more critical roles in certain program, and we're going to use that as a standard to determine what is needed for model evaluation. So given that, I'm going to summarize briefly for this portion of my presentation. So I wanted to give an overview for the models we have seen in new drug space. So essentially, we know that multiple modeling approaches are commonly used to support new drug development. And models can be applied to address various types of questions and different stages of new drug development. So this is what we call drugs or models are probably associated with different contexts of use. And we know that model development and relative decision-making is actually an iterative process. So we're always in a dynamic process to fine-tune our model and use the model to drive next decision-making. And some of the models may be reused in different development programs. And we understand the context of use is probably very critical to determine the model risk, which can be applied to select the needed activities and acceptance standards for model evaluation. So with that, we understand maybe sometimes we're facing the same question of interest, but the context of use and subsequent model risk for reusable model may differ from a single-use model for a specific program. Then appropriate model evaluation is needed. So with that in mind, I'm going to move on to talk about the fit-for-purpose program. So essentially, fit-for-purpose program is a regulatory pathway for acceptance of dynamic tools. Or like I said earlier, this is also what we call reusable models or model frameworks in drug development. And it really represents a joint effort, of course, across multiple disciplinary review team members and all stakeholders in the tool development. So by the end of the review process, generally what we do is we actually give a designation letter indicating that this tool is fit for purpose under several conditions. And this table actually summarizes what we have granted the applications so far. The first case is actually what we call Alzheimer's disease model. It is actually the disease model and includes a placebo and disease progression components. And also we understand this tool can be utilized to understand or to actually help the future trial design, because this tool actually includes a lot of components in the model, including the demographic information, the dropout information. And the second tool I wanted to talk about is actually is from Janssen and Novartis Pharmaceutics. It is a statistic model, what we call MCP mod. It has been determined fit for purpose for dose finding. And the third tool is from a professor from the University of Texas, MD Anderson. And he actually tried to share with us, and they can use a statistical model, which is called a Bayesian optimal interval design or Boyne design model. This is actually considered fit for purpose for dose selection. And then there's the fourth one is from Pfizer. They actually try to suggest that we can use what they developed, a so-called empirical based Bayesian Emax model for dose selection. And so those are the cases been granted so far. So in the next few slides, I'm going to walk you through about some of the key components in each of the submissions. Essentially, I'm going to focus on what is the context of use, because to us, just telling us this is a model by itself is not enough. We have to put in the appropriate context of use. So we wanted to talk about the context use and the evaluation approach that the review team has really conducted, to especially some of additional approach beyond the standard in order to understand whether the model can be reusable, and also the conclusion from each of the submissions. The first case I'm going to talk about is Alzheimer's disease model, which is a simulation tool to provide quantitative support in design and planning of clinical trials involving subjects with mild to moderate Alzheimer's disease. And it can be used for sample size calculation, optimal trial design duration, treatment measurement times, and competing trial designs and data analysis. The team actually look at the submission from different angles. The focus for the submission, again, beyond everything on the standard side, the focus for this submission is actually people really focus on assumptions it's been made for this tool, and also looking at the predictive performance of this model, and also what is the appropriate development programs. And also, by the end of the review cycle, the team actually indicates that the disease models are not intended to be a static constructor and characteristics. And we actually recommended the sponsor to continue revising this model given the new knowledge and the data they have. So this is it's a platform we consider is appropriate, but we also indicate that this platform needs to be revised with additional information we obtained. So the conclusion from the team is that the submission is actually scientifically supported and suitable for the purpose of aiding the design of future clinical trials in patients with mild to moderate Alzheimer's disease. And next one I'm going to talk about is MCP model. The goal for the MCP model is to serve as a strategy to explore and identify adequate doses for drug development. So essentially, this approach has two steps. The first one is so-called dose response signal, which was detected by using multiple comparison, and to select what is the appropriate model. And the mod step entails fitting the selected model to the data and estimated the target doses. So again, like I said, beyond the typical ways we evaluate the model, the focus for this review team is really looking at simulation studies, which is intended to demonstrate the benefit of using MCP mod and compare to other approaches. And also the team really looking at what we call generalizability and applicability of the procedure, and also the team try to identify what is the publicly available software packages. And the conclusion from the team was that it is scientifically sound, determined, fit for purpose in outline context. And the third example I'm going to talk about is the Bayesian design for the maximum tolerated dose. Again, as we identified earlier, this tool is really trying to use to identify the maximum tolerated dose based on phase 1 dose finding trials. And of course, in this case, the MTD, maximum tolerated dose, is defined as a dose with a dose and limiting toxicity probability closer to some sort of pre-specified target value. In order to assess this tool, the team really focuses on the methodology and also and try to understand what is the mathematical or theoretical basis to derive the Bayesian design, and try to identify the applicable scenarios which the Bayesian design is suitable, and also try to assess the simulation scenarios with understanding of some of the limitations, and also try to focus on the different software implementation of the Bayesian design. And also, the team actually finds out there's something that needs to be improved from theoretical side. So essentially, they give this suggestion to the applicant. So essentially, the applicant agreed to have a revised form of the Bayesian design, and also they also identify what is the appropriate usage for this Bayesian design. So the revised usage together with the new theoretical form has been granted as fit for purpose. The fourth one, which is the last one, is the Bayesian Emax model. It is a statistical methods and supporting software that are proposed to improve the design and analysis of clinical trials, whose primary purpose is to characterize the relationship between efficacy and dose to guide the dose selection for future development. So in order to really looking at all the designs, the team actually focused on the assumptions, looking at how it's been applicable compared to other alternative designs. Also, the team really focused on the suitability of the goodness of fit statistics, especially talking about how this model can be predictive for some of the outcomes we're looking at, and its applicability. And also the team also focuses on the simulation studies and how this can be generalized into all the different scenarios we commonly see in drug development. And also they look at additional data and sources in our package which can be used in public. So the conclusion from the review team is that the review team finds that the proposed empirical base Emax model, including the goodness of fit statistics, can be designed fit for purpose under different conditions as laid out in later here. So just, with that, I'm going to conclude my presentation today. So the message I wanted to deliver is the following. So we think the Fit for Purpose initiative is an established regulatory pathway to evaluate dynamic tools, or you can call this as reusable models in new drug development. So in line with the common features seen in the models applied in new drug development, the models considered fit for purpose should be defined under a specific context of use and assessed by appropriate model evaluation approach. And the needed model evaluation activities and standards may differ from models developed for a single development program, and model structures and parameters are generally not static. And even though the model is considered fit for purpose, it does not preclude the use of other modeling approaches to address the same question of interest. So with that, also, I wanted to add some of our initial thoughts for how this model master file can be potentially applied in new drug space. Again, doesn't mean this is our position now. We're just using this as a starting point to generate or to accumulate more response from your side. So we think the initial context of use of these models is to support the bioequivalence evaluation in life cycle management of NDAs. For example, as we had some discussions in the roundtable yesterday, and one scenario I can think about it is, let's say an innovator needs to change their manufacture sites in multiple times. Maybe there is a value to develop this model master file because they wanted to really support their manufacture sites changes. So that could be one scenario we can use this model master file. And we wanted to emphasize the model, including their parameters, is expected to be used repeatedly by the same or different sponsors or applicants for the same drug substance and the route of administration. And the model structures and all parameters are built across all indicated populations through sufficiently large database in the drug development program or different programs. And there is an anticipation to not to change the model structure or parameter values in a meaningful level with new data or specific context of use when the model master file was initially determined to be acceptable. And there should be sufficient information to evaluate the model performance to the target context of use. With that, I wanted to really thank a lot of individuals who helped me to think through this process and also helped me with the preparation of the slides. Last but not least, I want to thank you for your attention.
I thank Hao for the great discussion on the dynamic setting of model tools and the diverse context of use of those tools for new drug development and regulatory decision in new drug development setting. And our next speaker will be Yuching, and she will discuss current practice in model evaluation and assessment of PBPK model reusability in new drug development. Welcome, Yuching.
Thank you for inviting me for this wonderful meeting. And so today I am going to discuss something related to how the PBPK model was reused to support the new drug development. Everybody is talking about the arrow. Okay, here. So I don't need a disclaimer because this is a topic we have not presented in other place. So first, let me open with the overview of the PPK analysis in the office submission status in the Office of Clinical Pharmacology. So basically, the PPK modeling and the simulation has become a stable tool used in the drug development. And the slide show here, on the right-hand side is the submission summary by the year. as received by Office of Clinical Pharmacology. So as you can see, we can observe an increase over the year in the PPK submission in both small and large molecular, which means the NDA and the BLA. So the bar figure is basically showing how many submissions we received per year, that including both supplemental and original NDA and the BLA. So it is estimated that between 20 to 30% of the newly approved drug now including the PPK analysis as part of their regulatory submission package. So on the other side, the pie chart illustrates the distribution of the PPK submission in terms of the application area. So PPK modeling has been a critical role throughout the whole pipeline of drug development, and it can be used to evaluate the effects of various intrinsic factors and also extrinsic factors. So as you can see from this pie chart, the use of PPK modeling is particularly significant in drug-drug interaction assessment. So it accounts for about 60% of the application. So now let's jump a little bit talking about the PPK model evaluation. We call it the fit-for-purpose model evaluation, but this is just a concept. It does not relate to the fit-for-purpose initiative that Hao described previously. So basically, we understood that development of a mechanistic-based model often requires substantial resources. This investment reflects the complexity and the precision this model must have in order to effectively simulate the biological system and the drug's ADME property. So basically, it requires the integration of the knowledge across different domains, biologic, chemical, and pharmacological. And I think I don't need to go through each one of them, but I do want to emphasize the contribution of the PBPK platform, because it did provide, in a way, integrated tool to summarize and integrate all the knowledge. And but for this platform, one critical way is under this unified framework. It provides some feasibility to assess the model structure. So it can the platform usually need to be need to be robust enough to handle the new data acceptable to the emerging insight and capable of involving with the ongoing reuse, research and the development. On the other part, regulatory acceptance of a model or its prediction is based on the modeling and the simulation. Based on the modeling and the simulation result is determined by their appropriateness for the intended purpose. That implied even when the same analysis is used, validation and acceptance criteria are contingent upon the overall model risk or risk posted by the model. Such risk encompass the model's significance within the entire body of the evidence and the clinical risk associated with incorrect decision making. So therefore, depending on the impact and the associated risk of the model, regulatory expectation can differ at various stages of drug development. So let's go through an example about the PPK review focus at the NDA stage. So as showing, this is a flow chart, and the first step is to clearly identify the context use of this PPK model, how the model result will be used to support a specific labeling claim or recommendation, and what is the model risk. And then the second step is to review the model adequacy. So from this step, the reviewer will evaluate the mechanistic assumption in vitro data, in vivo data set used to support the model development and the validation. So basically, what is the outcome? So if the model is not accepted, then the PPK model data will not be used or included in the label, and there may be post-marketing study or PNC needed. But in some cases, the model can be refined by incorporating additional mechanism or collecting additional data set to refine this existing model to confirm certain model parameter. So in that case, the model might be accepted after refinement. So if the model is accepted, then the simulation result can be used to support the labeling and our labeling information. So it depends on the review objective. Alternative mechanism or dosing regimen can be simulated to understand the magnitude of response under different scenarios, such as the polymorphism. So to put this idea into the context, let's look at the one specific example. So this is the one common question we heard repeatedly during the presentation is how many clinical validation or how you declare a validation is adequate in order to support the acceptance of a PPK analysis. So here I just want to use this example to illustrate the scenario often showing in a DDI NDA evaluation. So first of all, the scenario here is the model is going to use to support if what is the dosing recommendation for a drug when given with a dual CYP3A and a CYP2C9 inhibitor. So on the front end, the drug already have a certain level of the clinical study which can inform the individual pathway, the impact of the individual pathway by a pathway-specific inhibitor. But however, in order to use the model, not the clinical finding, to support this analysis, we implement the PPK model. By using that, we want to use the inhibitor PPK model to inform, to quantitatively estimate the contribution of that specific pathway. In this scenario, will be CYP3A and the CYP2C9. So in order to quantify or validate an inhibitor model for CYP3A, you need to have a good sensitive substrate, pathway-specific substrate in order to verify this pathway-specific inhibitor. So similarly, for the 2C9 pathway, in order to verify the performance of the PPK inhibitor on the 2C9 pathway, you also need to have the sensitive 2C9 substrate. So basically, if you count everything, basically at least just for this scenario, and we're only talking about using the single substrate model to verify a single pathway inhibitor, we're talking about at least 8 drug PPK model needed to be validated in order to to support this PBPK analysis. And for each of the PBPK model we included here, we need to have an independent verification and assessment. So this slide illustrates the difference, but the dependency between the regulatory acceptance of a PBPK analysis and the reusability of a PBPK model. So How had discussed to evaluate any modeling assessment, we implied the model risk assessment framework. So basically that means the adequacy of a PBPK analysis submitted in a regulatory package is determined based on model risk and the totality of the evidence. That means the review team consider if the PBPK analysis is adequate, if it can address a specific question for a particular case through the modeling and the conclusion. But when a PBPK model is considered reusable, we do expect that the model can support a particular predefined context of use across multiple programs. So the model's assumption and uncertainty should be well documented, and the model should be substantially validated with multitude of the clinical data relevant to the specific context of use. So what does that mean? That means when a PBPK analysis is accepted to support a regulatory decision, it does not automatically imply that every model and assumption included in a model or the analysis is guaranteed to be reusable for the next application. However, if a model is considered reusable for a specific context of use, we consider that a reusable model should be suitable to be used in another PBPK analysis to address any question where this specific context use of this model is relevant. And I want to reemphasize now is we do not yet have an official program to formally recognize a PBPK model as reusable. So which means that currently we do not have a PBPK model go through the fit for purpose initiative. However, reuse of a previous validated PBPK model is pretty common. So as we show in a previous example, to support a PBPK analysis, you do need a suite of the PBPK model, not talking about the background information, talking about the background framework or model structure, etc. So basically, you do need a really rich body of the information to support a PBPK model analysis. So therefore, reuse of a previous validated or verified PBPK model is common, especially when evaluating the DDI liability of a new drug. So, last in our NDA review, several PBPK models for the sensitive substrate of a specific enzyme pathway or the inhibitor or inducer model for the specific pathway are commonly reused after it has been sufficient and repeatedly verified with different source of the clinical DDI data. So, so in that case, when this model was employed in a PBPK analysis, reviewer typically perform modest or minimal model evaluation. However, I still want to emphasize the review activity for a given PBPK model or PBPK model analysis depends on its context of use. And so therefore, we still require the documentation of the model development and the validation is needed to be included in each of the for each of the model and need to be included in your submission. And the reusability of a previous verified model need to be assessed and determined during that review for that specific application. So I want to use a case example. To show the what we are saying to put into the context. So the scenario I have here is a workflow to evaluate the OAT3 transporter mediate DDI. So OAT3 is a basolateral transporter. So the background is in terms of clinical DDI data, the clinical study find the OAT3 inhibitor probenecid increase the exposure of baricitinib by twofold. So the concern will be how about the other in vitro identified OAT3 inhibitors such as ibuprofen. Even though based on the in vitro data, it looks like the ibuprofen might be a weaker or less potent OAT3 inhibitor than the probenecid. But how does that translate into the final labeling? So, so in this submission, the sponsor on the PPK part, they implement the mechanistic kidney transporter model to identify to verify the OAT3 mediated the uptake flux based on the in vitro data. And then they verify this PPK model with the clinical PK data and the DDI result. And through this framework, they want to predict the substrate exposure under untested DDI scenario. So basically, try to interpret from the a stronger inhibitor probenecid to a less potent inhibitor ibuprofen. But the one thing I want to especially point out the acceptance of this submission is because in addition to the PPK model, the sponsor, the applicant actually developed workflow in terms of the in vitro assessment, which means they verify their in vitro assay by testing the cases in the blue column. So basically, the same in vitro assay was used to verify the in vitro to in vivo performance in a separate study that is another compound. So after the in vitro assay was verified, they parallel move this in vitro assay to the baricitinib. That is why this development of the in vitro assay, the rationale they can use the in vitro verified parameter put in a model prospectively agree with the observed data. The whole framework move into the baricitinib was also showing a successful. That's why that is one critical underlying rationale for acceptance of this submission. So in a result, the model, the simulation in the labeling, the simulation was used to support the labeling, and it was declared that we consider the other OAT3 inhibitors, such as ibuprofen, will have less inhibition potential. So now the question will be, Okay, so what happened to those PPK model? It was accepted in your NDA review. Does that mean those model can be considered reusable for the next application? So basically, we can look at it one by one, and so to get a consensus when we're talking about acceptance of a PPK analysis and the reusability of a model. So in terms of the baricitinib model, what is the likelihood for this model to be reused for a similar purpose in the next application? We consider the likelihood for this baricitinib used as OAT3 substrate, the likelihood is high because the mechanism is well characterized, etc. But however, the FDA reject the model for this baricitinib PPK model used to support the RA population because the sponsor was implement the scientific rationale to declare that based on literature and some scientific information, the OAT3 expression level will be less in this disease population. However, we do not have the clinical data to support this scientific rationale. So even though the baricitinib model as an OAT3 substrate in healthy volunteer, the likelihood to reuse this model for this specific purpose is high. In the clinical practice, we reject the model to be used for the RA population. So how about for the probenecid model as an OAT3 inhibitor? We also consider the likelihood for reuse this model for this specific purpose is high. However, we do need to know some specific underlying assumption and condition related to this pathway. So that means we identified the substrate independent inhibition parameter, which means the current parameter assigned for the baricitinib PPK model. Is that parameter considered substrate independent? Can we apply that to all across the board to any OAT substrate? That piece of information need to take into consideration when the next review case comes up. So how about the other two inhibitor model? So we consider the likelihood to reuse as the model as an OAT3 inhibitor is less and need to be determined case by case because basically the clinical study showing for these two additional proposed inhibitor, they did not have so-called clinical significant response with the OAT3 substrate. So therefore, we do. So if the sponsor applicant claim, oh, I conduct study with ibuprofen, I did not observe any meaningful change in the plasma PK. Does that mean my drug can be declared as not a OAT3 substrate? We consider for that purpose, this model basically is not suitable. So here is some comment in terms of the reusability of the PBPK model in the new drug development consideration. So basically, when we consider the reusability of a PPK model, I think a thorough initial review is very critical to effectively reuse a model. And I need to point out this initial review could be resource intensive. And in order for the next user to actually know how to reuse and what is the details they need to be pay attention in order to correctly reuse or partially reuse this model. The documentation or the focus or classification you may put into to describe the reusability of this model need to be very precise and but also need to be consensus. And we also need to consider when in certain scenario, re-evaluation of an existing model can be triggered by several factors, which we discussed those scenarios extensively in yesterday's discussion, including change of context of use when the model was used for different purpose. Certainly it will trigger a re-evaluation. Also, if they have the new science and technology update, certainly it will trigger evaluation. And one more point I want to point out is, okay, I have a model which has been previously verified, has been accepted in the NDA submission. And does that mean this model can be used, and what is the scope and the range scenario I can reuse this model? I want to point out that to navigate and understand the complex information to determine the reusability of a given model could be similar to compared to determine if the model is fit for purpose, fit for this regulatory purpose. The reason is the documentation for the model to describe this model is likely outlining, emphasize what the model has been used for. But it might not including the very clear and definite information about where the model should not be used. So as a result, in many instances, determining whether a previous approved model is suitable for a new scenario require as much effort as reviewing the model for a specific objective. So that kind of effort to go through very detailed information to think about if I can repurpose this model, reuse this model for the next application, the effort to put in this is kind of similar. So basically we need to justify the upfront resource we want to put in to document or to develop or establish the reusability of a PPK model. Thank you very much.
Thank you, Yuching, for the comprehensive discussion of the PPKK evaluation and model reusability. So next one will be a presentation from Joga. He will discuss the potential of repeated use of PPK model to support a BE study.
Good morning, everybody. Thank you for the opportunity. Let me see if I can get this slide thing right. Modeling is easy, but operating this could be challenging. So I wanted to take a couple of minutes in the beginning to set my perspective, and I want to share that because we need to appreciate that as a starting point. First of all, 10 years ago, 15 years ago, would any of you in the audience expected to meet to talk about scaling up modeling activities? No. Yeah, one guy. We're talking about drug development. So you can see how the agency, the pharmaceutical companies and academicians, sure, moved the needle so that today we're talking about scaling up of activities, modeling, application of modeling to foster public health. So this is a good thing that we are here to talk about. The second, and I would like to acknowledge the efforts of Office of Generic Drugs and Office of Clinical Pharmacology and other companies who have invested in this and moved the needle. So it's a collective effort. The second important thing, why I think Office of Generic Drugs is fostering this idea about model master files. Let us forget about model master files for a second. The challenge that they're trying to solve, in my opinion, is the following. For every non-complex dosage form, do you know how many generics are approved on an average? Not one of you? Okay. It is about 6 generics for every brand non-complex dosage form. Do you know how many generics are approved for complex dosage forms such as long-acting injectables? No? It's not even 1, it's 0.6. That causes a disparity in healthcare in our country. Patients cannot afford these promising treatments because of the price. And Office of Generic Drugs has this public health burden to find a way to facilitate more generic approvals such that they tackle this disparity. And one of the initiatives that they are proposing here is that, is there a way for us to reuse or make these quantitative approaches more available for generic companies, because not all generic companies will be able to create departments to build these models. That is the reality, and they have to solve this problem. So I would like to urge all of us to keep those 2 things in mind, why we are here. And if MMF is not a good idea or they have an alternative idea, I suggest we pick up a weapon and stand opposed. Propose something else and we can talk about it. But here we are to talk about model master files. Let us give it a chance. And what I'm going to do, I'm a country clinical pharmacologist, that's who I am. So what I would like to do is to take a Socratic method to probe some questions for us to think about, so that it would be successful or it is vetted thoroughly and it could have the best chance to succeed. My intention is not to paint a negative picture. Now, I would like to contrast MMF and DMF. DMF is something we know, drug master files. Let's start there. What is the purpose of drug master file? It is to protect confidentiality, proprietary information, streamlining approvals, flexibility and efficiency, and regulatory oversight. To me, based on my own experience, DMF's single most important value add to the agency is that it cuts down on site inspections, which is a very intense, resource-intense operation for the FDA. It also cuts down on review time for the same manufacturing site to be, reviewed, the material to be reviewed over and over. From a sponsor's perspective, it is much more, it gives a flexibility and efficiency because they can go to a manufacturing organization who has a DMF approved by the agency and then not worry about the quality of the product before they can release into the market. So that's drug master file. Now let's talk about the topic that I was given is about PPK models. So I'm grateful that they confined my talk to PPK models. And I will present two scenarios. One is what we can call as biocompatibility, meaning within the same brand company, they change the manufacturing site, or clinical to commercial formulation has changed, or for life cycle management after post-approval. These cases sometimes, and especially for complex dosage forms, a BE study is required. So in these cases, I'm going to call this, the sponsor as sponsor 1. I'm going to introduce sponsor 2 in the second scenario. So this is the brand sponsor. And currently, the process is the following. You have an original submission that gets the regulatory review, and in terms of the model, and followed by its application, meaning whatever that intended purpose was, that model will be applied to support that regulatory decision, okay? Then let's say the same brand company comes back for one of those 3 reasons I showed in the previous slide. Then the scientific review would take place again for that supplement, and the agency will decide on the ask by the sponsor. Now, the questions that we might want to ponder are, does FDA repeat model review for each submission? Again, in the context of POP-PK model, they would, but I think it is reasonable to assume they would review these model to see if there are major or important changes from the original model without having to assume they would start from scratch. Then does the change in reviewer affect model evaluation? This is a question I have for sponsors. Have you ever encountered a scenario where that if model, the same model was submitted a second time, did you get different questions from the agency, assuming the context of use has not changed drastically? So it would be good for us to understand and make a case for ourselves. What is the bottleneck? Is it the model review or the application review? The distinction I draw is model review is a technical piece, scientific, but application review is not. It is a drug development question. In the context, should I approve this drug? Should I give this labeling statement? Those decisions are never based solely on modeling. It has to be, judgment is always involved in that. And so if we can identify that bottleneck, it'll be useful. Now, let's assume we have model master files. Then the proposal is that when there is a second submission, we're still talking about the same brand company to the brand company, within the brand company, meaning they have access to all those data, safety, efficacy, and quality. Then you can skip this scientific review in the second cycle, that's why I strike it out, and straight go to the application. So that still has to be reviewed because now it's a different ask compared to the original submission. That's the purported advantage of model master files. And here, there is no need to repeat the model review, reduces between reviewer variability, and so the question then becomes, and this has come up yesterday as well, is it possible to dissociate the model review from the decision review? And I think my previous speakers today alluded to that as well. Having said that, still a scientific review of the model is inevitable. We still have to go through that before we can make it contextual for that purpose. So there could be an advantage if there is a very complex model that we don't have to revisit the review every time. Now, with MMF, sponsor can potentially obtain FDA review before the NDA BLA submission. Now, this is an important point I want us to think about. Let us say that the brand company, sponsor one in this case, is developing a complex model. Then they don't have to, and there is a, let us say, a very important regulatory decision that is impinging on that model. they can, decrease the uncertainty by submitting a MMF early on during the drug development. And this is something we'll have to talk about, which is it's a logistic regulatory thing, is can a sponsor submit somewhere before the NDA BLA submission and get a review by the FDA? That could be helpful for the sponsor to kind of alleviate surprises when they really submit the NDA and BLA. But you can argue with me, why can't I use one of the available pathways, type C meetings and such for that purpose? But a dedicated model review might be, it's not a bad idea in my opinion. So let's think about it. Do we have experience with MMF submission so far? As I gather, probably not. But you have to start somewhere. So we may want to discuss the data standards. Data standards are set. I don't think that needs to be revisited. A model file format, that's also pretty much set. There is a way we figured out over the years how to submit these files, even if they are proprietary files. And do they need to be software agnostic? So again, going back to the original impetus, public health impetus for us to consider something like this is to make this science available to foster generic drug availability to patients sooner, okay? So with that, software agnosticity is an important matter we may want to talk about. And then how do you want to do the report? We know how to submit these components already. Now let me switch over to the next scenario, which is a 505(b)(2), meaning there's a second sponsor involved now. So the sponsor 2 is the one that is submitting the 505(b)(2). And the key question in 505(b)(2) is reference to test bridging of efficacy and safety. So that is it's a gray area, I will say. There are no clear guidances and so on 505(b)(2)s, which I actually appreciate. So there is flexibility. And modeling has lately been playing a great role in 505(b)(2), supporting 505(b)(2) applications. Now, here, the first step is the sponsor 1, there is an approved MMF. Now, here is the second part is where, again, it goes back to IP, legal, and all that. We can talk about it. Right of reference to sponsor 2. I'm calling it right of reference. The right of reference is inevitable anyway for sponsor 2 to procure from sponsor 1 for 505(b)(2)s. But here we're talking about a model which I will have to think of how that is going to be feasible, or it is also possible that instead of sponsor 1, somebody else would have this MMF. But is that possible when the proprietary, I'm distinguishing between confidentiality and proprietary information, resides with sponsor 1, not the third party, okay, unless everything in the model is derived from public sources. Possible, but something for us to think about. And then the sponsor 2 files 505(b)(2), and then there is a regulatory review focusing only on the application rather than the modeling part, okay? And this is more, contextually, it is more, I think, relevant for us to consider complex models. I'm not talking about IV bolus. I mean, IV bolus, you don't need a BE study. But something very simple, one compartment model, we're not talking about those. Now, here are some practical questions for us to ponder, which are, who submits the MMF? I made a note that there are at least 2 possible entities who could do that. Sponsor 1, I don't know if there is any motivation for the brand company, the original brand company, to put their model up for grabs. But it is possible there's a third party who might do that. How does sponsor 2 access MMF? Would they run their model or simulations, predictions by downloading some kind of a file and running it on their computer, or they would have to go to a third party? Yesterday, I met somebody in the audience who said that's what they do. You know, they get the data from the sponsor, and they run the model predictions as a service for immunogenicity predictions, and that would go as part of a report to the FDA. That is a reasonable model. And does sponsor access model data? That gets tricky. How do we rerun model for test using MMF? Now, this is an important question we will have to talk about. So the original model, my assumption would be that it contains only the reference data. What do I do, or what does the generic company do to supplement that model with the test data? Do they have to rerun it, the model estimation, or do they have to, because there is a predictive nature, like PBPK models and such, it's a predictive, there's no estimation, well, legitimately. But if you're talking about empiric models, like the topic I was given, POP-PK models, those are empiric, meaning you'll have to estimate. Do you re-estimate by combining the data, or you fix the model parameters and re-estimate for the test? And, you know, and we get into all that scientific debate about it. And the sponsor to perform, so this is the part where I'm going to provoke a little bit. Now, if we have the reference model accepted by the FDA, can the generic company get a waiver for studying the reference in the BE study? We can talk about it. Then will the review time be shorter? I'm not sure. So again, I want to remind us, to me, review time is not the issue, honestly. It is the availability of science, like Rob and Liang and Lucy elucidated. It's the availability of science to foster generic drugs available, products available for patients. So review time could be an added advantage, but the availability of science itself is a challenge, in my opinion. So with that, I wanted to thank the organization, and I hope my points are useful for us to think through on this important topic of MMF. Thank you very much.
Thank you, Joga, for the great discussion. Now we have more questions. It's perfect for panel discussion. So now I'd like to welcome my panelists on the stage. And as you can see, we have 3 new panel members. So before we start, maybe we can give 1 or 2 minutes to each new panelists to give their thinking about this session, overall thinking. Maybe we can start with CJ. Sorry.
No? Thank you. Is this on? Yeah. So thank you very much, Jiang, and all the speakers of this session. I think it's been very interesting for me to contrast the difference between new drugs and also the generic space. It's really been eye-opening to me. I do see differences and similarities in terms of what we could do as a modeling and simulation community in terms of this master model file. Model master file. And that's a point I actually wanted to make, is that it is a model master file and not a master model file, right? Master model is one thing. We're not talking about one model to solve everything. We're talking about multiple models that are iterative as we learn more information, especially for the new drug space, that information is incorporated, and then we apply that model and reuse the model for different applications within the context of use. So for that, I'm really open to the discussion today. I have an open mind regarding this, and so I will look forward to the other panelists' opinions.
Thank you, CJ. next one, Martin, you can go ahead.
Oh, yeah. Yeah, well, yeah, thank you. Thank you to everyone for the very nice discussion. And just reflecting on some of the things that Hal discussed, thinking about the Fit for Purpose program, as well as what Yuching had talked about, the framework for assessing and evaluating the PBPK models, and then Joga, some of the questions that you brought up. It brings to mind that the model master file approach could be useful for as a way of formalizing formalization of our understanding of a model or methodology, including the scenarios under which the model has been evaluated, scenarios under which the model may be applicable but has not yet been evaluated, as well as scenarios where the model may not be applicable. So this could serve as a very useful framework for formalizing that understanding.
Okay, to our KOL Liang, please. In the new drug space. Okay.
Yeah, so thanks, Jiang, to give me the opportunity. And also thanks, Joga, for doing the kind of understanding speech for generics. So with that, I also want to mention something about new drugs reciprocally. For new drug, so if we look into the history, so we always advocate novel methodologies to cut the cost, cut the time. That's been the initiative for many, many years. Modeling have been thinking is one of the innovative tools that are gonna be a game changer. If we look at the data, so for the cost, even adjusting the inflation for new drug development, certainly we are facing a high barrier. So the cost actually has been increased to on average over 2 billion. I think that's the correct number. Regarding the development cycle, so I have reviewed like a Insight report last year. It has been stagnant. There's no time cut. So in that regard, should we introspectively looking at us, do we have pressure? So we have been advocating modeling simulation. Has that really, how much I think that contributed to the game changer? So my point is we become stagnant and we are not, really pushing hard with new technologies, new regulatory pathways together. So we may not be making up to the expectations. So model master file is one of the things, as mentioned yesterday. I think the time is ready for us to think some new ways to really make the program more efficient and as a way to cut the cost. I will stop here. I will welcome more thought from others.
Do we have any question for in-house? Okay, go ahead.
I have a question.
Go ahead.
Annie DeGroot from Epivax. We're the immunogenicity modelers. One question is to go back to the idea of submitting the model master file separately from an ANDA submission, for example. I heard that during your talk, and I was unclear about how that could actually happen. There's also a question in the chat about that. Perhaps we could hear more about that. And my second question is, how much detail do you need on the models? Because, are we submitting the code, or are we submitting the description of the model rather than the actual code? I would hope it would be the latter, not the former.
So maybe we can refer this question to Joga first, then how?
These are the views of the FDA, not mine. Just kidding. Just kidding. My thoughts are just, I've seen both sides. So the way I would define MMF is going to be that this is confidential and proprietary information pertaining to the model structure, equations and such, scripts, modeling process, and or the model qualification for the intended regulatory decision support. That is how I would define MMF. Now, for when we submit, it doesn't matter if it is FDA or somebody else receiving this, because we're all scientists, we think alike. When I have to make a decision about that, I would need the data, I would need the scripts, I would need to be able to reproduce it. So you sending only a description is not going to cut it to me at all. The reason is, think about it. If it is only for one purpose, that drug, the sponsor is going to come with empiric data as well, and maybe supplement some of the thing with the model to support approval or labeling decisions. But now, if a MMF is accepted by the agency, and now half a dozen companies are going to come, I want to be doubly sure. So I have to pressure test it. Beyond that, the reference or whatever you use to develop the model, so I would need all of them, no question.
Just to clarify, your question is about ANDA or just NDA or BLA submission? It's about ANDA, right? And maybe I'll refer back to you.
Yeah, I think AN, right? So welcome to, appreciate your presence in the meeting. Actually, we kind of mentioned in immunogenicity, in silico immunogenicity assessment tool development meeting, then we kind of mentioned this workshop too, and thanks for coming. I haven't managed to talk with you yet. I think for immunogenicity assessment is very, very important topic, but currently is not in the mainstream of the modeling topics yet. However, I think it's a very much overlooked important issue. If there's a standard way, for example, either for new drug or generic drug, that we can use in silico tools to assess the risk, and then probably have a decision tree, based on the tools. The immunogenicity risk reached to a 13 threshold value. Above that, we need in vivo, in vitro testings. If below certain level, then probably in vitro only, or even below, we are very confident with something, then even we can trust in silico predicted result. That ideally, that could be the case. Currently, we haven't done anything in that area yet. Regarding the model master file submission, if your model you think is mature enough, then we would expect everything. That's the beauty of model master file. So even you have the code submitted, it's only accessible to FDA. So we need to duplicate the result, especially at the beginning. And I think FDA would respect the confidentiality and if there's a concern, but that will certainly help you standardize and broadcast the progress you made from your company regarding in silico immunogenicity assessment. I'm not sure if I addressed your question.
Okay, go ahead.
Yes, I'm Marcus Fridén, AstraZeneca. I'd like to understand a bit more about the relation between the model master file concept and this fit-for-purpose program. I think they both address similar opportunities, but whereas the fit-for-purpose program is sort of described to address dynamic models that evolve with a greater speed, the model master file is intended for more static models where you don't intend to make changes so often. That's how I understand it. But in my experience, let's say in the new drug space, it's not that black and white. I feel models models evolve everywhere when the understanding increases and as you get more data. So even in the sort of life cycle management space where I operate, I now feel uncertain which of these concepts or frameworks are kind of more useful to me when I'm kind of thinking ahead about the changes that we will, or improvements, refinements that is coming with our models.
How do you want to address that?
Maybe you can start it. I think this is a good question. Well, the Fit for Purpose program has been existing for more than a decade now. So, like you said, we use that in the new drug space to address some of tool development needs. And like you said, it's the way we try to handle it, we look at it more like a dynamic process. So we allow, and actually we encourage the sponsor to say fine-tune the model with the latest information, and also use the fine-tune the model with the better understanding of mechanisms, fine-tune the model with new data. So that is what we actually stated in the Alzheimer's disease model. That was the first Fit for Purpose program we've been through that program. So, the model master file is a concept that we recently, interacted with our OGT colleagues and try to bring in and harmonize. The way we try to do it, as laid out in our presentation, is that we might think there's a space we can also incorporate that content into our pathways for models where we think it's more matured. In that setting is, like I said, the structure's been set up, the parameters been estimated, the program is almost done. 10 years of development program is almost complete. And by the end of the day, for example, one example we laid out is in the new drug space. Maybe there's more from the generic application also, but at least in the new drug space, as you hear from Dr. Gobburu or my presentation, is that, let's say, for example, you wanted to make some manufacturing site changes, or you want to expand your manufacturing sites to multiple, let's say, CROs to help us to expand the supply chain, or you're facing shortage issues, so you really needed to expand the supply here. That's where we see the model master file, which we have a more defined structure or more defined parameter space. Then everything's been settled, we can use that to justify that we can quickly, support the expansion to the new manufacturing site, where traditionally you might need a BE study or something like that. Now we have an approach here. We have, more potential to expand the capacity. And those are the angles we think, but we also want to welcome the discussions from the audience. Just try to understand, where do you see the usage of this? And so that we can best use or maximize the value from model master file, and also try to maybe carve out some of the usages so that we can really benefit and without getting into some of the challenges we also discussed earlier and yesterday and today. I'm just saying from the new drug side. I don't know if Liang has something or other panelists want to add anything.
CJ has.
Yeah, I wanted to comment on, one area that has been offered as ripe for this type of modeling approach or for standardization, and where the FDA has actually been quite progressive, is in evaluating disease progression models. And those are models that we can use over and over again when we have a therapeutic area where we are very invested in multiple compounds coming through or combinations of compounds coming through. So that's an area, I think, where if we can have a standardization of what is expected in a submission in a file, and then also for each disease area. Another topic I wanted to hit on was the immunogenicity framework, because that is an area that's actually quite active in the new drug space. There are several companies, commercial companies and pre-competitive consortia that are tackling this issue. I do think that is an area, anything with immunogenicity or safety. There's been many initiatives in the mechanistic modeling space with SIPA for proarrhythmic assay evaluation. So I think there's many opportunities here for standardization and harmonization for areas where we have a lot of commonality. So I think that's also. I also want to just, with Liang, your comment about accelerating drug development through modeling and simulation, I'll point you to some publications that, our company has invested in MIDD for many, many years now, and we have significantly reduced our drug development time, and we have looked at the root cause, and we can really point to modeling and simulation approaches for doing that.
Thank you, Siri. So I can make a comment, then there's a relevant question from Dr. Karpak before he left off for a meeting associated with the disease progression. So my comment is that in the model master file realm, I think a generic, we want to take advantage of the cumulative knowledge and the data and the model that being developed through the new drug development cycle and post-marketing stage. I think in new drug, I think the nature model is mainly for benefit risk profile analysis for generic. So we are more towards drug deliveries. So if there's a formulation change, will there be an exposure change at the site of action? So most of the cases are systemic exposure, but there are like 20 to 40% products. We want to see if the systemic exposure reflects the local drug exposure, et cetera, et cetera. In that regard, we are more open-minded to have a bigger scope for model master files. Yesterday, we talked about a product API specific model. We kind of discussed the methodology, discussed virtual organs, and if, certainly we welcome the audience to voice your opinion, even for new drug. If I believe Hao and Jiang and everyone in the new drug side would love to hear that. So I think we are more taking an open-minded approach to take advantage of the knowledge and the experience gained in the past, based on the risk and based on the science. That's what we are promoting from generic side. So before I think there are some question, but before that, there are associated question from Dr. Pak. His question is, can you see of employment of a highly data-informed disease progress model as a control group in a registration trial? For example, comparing a single arm new drug treatment arm with outcome from vetted disease models. If there's any comment in response to that question, it would be great.
This is really a $5 million question. There's no easy answer for that. It really, I would say, it really depends. When I say depends, is if it's a common disease and there's no challenge to enroll patients into, for example, placebo arm or control arm, I would say there's some challenges, at least for now, to directly use this model to generate the so-called virtual control arm. But there are some potential usages we're actually trying to actively exploring at this point is really get into rare disease area, where sometimes you see those disease progressions are pretty rapid. And even patients with just a placebo treatment is sometimes gets very unpleasant outcomes. So that is one scenario. And it's very hard it's very challenging to enroll patients into the study. So once you have a potential therapeutic advantage for patients, you really wanted to get them to patients, not leaving them out from just getting into placebo. Those are the areas I think where I can see there's a lot of attempts to see, can we actually apply, at least to get some information from natural history studies and disease models to inform what would be happening if somebody actually get the placebo arm. And you can see, for example, in the cancer space, and sometimes it would even allow single arm study. So what does that single arm mean? That means we trust the disease progression observed from the disease general, the progression from the past experience. If the signal is large enough, we think it's going to overcome what the uncertainty comes along with the natural history studies, or which is the sources for some of the disease models. Then it might be a benefit for us to make the argument or to support that the drug is potentially efficacious. So there are areas, but there are still a lot of challenges technically. So, but I think it's not directly to model master file, but that's something we're actively exploring that too.
Joga, you want?
Just a quick comment. But using any which method, including disease progress models as a potential control arm, is still okay in the early development phase where you want to support go/no-go decisions, dose selection, and so that is within the realm of sponsors' responsibility.
Yeah, we have all kind of discussion regarding this virtual control arm, even for the organ impairment study, whether you need the health subject. There's a lot of discussion there. I also want to hear from our statistics, his opinion on these topics as well.
Yeah, well, it's certainly a very interesting and important question, and to understand how the modeling would play a role and how we could how we would quantify the uncertainties in those kinds of situations and what data would go into that. It's a very interesting question, and at this point, it's certainly something for exploration. I don't see any easy answer at this point.
It appears maybe we are running out of time, so maybe we can discuss this. One more question?
Yeah.
Okay, we also have a lot of interesting questions online, so I don't know whether we have time to address that.
Well, let me just zip in. First of all, I want to thank the moderator and the panel. It's an excellent panel, excellent talks today. Really enjoyed them. I'm Jim Bosley. I'm from Nova Discovery. Dr. Zhao, I believe, mentioned that the goal of model master files would be to save time and money, among other things, and decrease uncertainty. I'm in the QSP space, so I'm sympathizing with the reviewer who has been used to getting a 10-compartment, 5-compartment model with 20 parameters, and now I give them a 300-compartment model with 500 parameters, and they're looking at me and saying, "What the heck can I do with this in 6 months? Get out, leave me alone." So that goes to what Joga said about pre-approving models. And so question one, and I have two questions. Question one is, would it be useful to have an industry standard QSP model for specific diseases that is pre-approved, that everybody can use, that the FDA would endorse? Because in that way, if you're dealing with counterfactuals in early development, you need a causal model, and you're not going to curve fit your way to predicting stuff that's never been tested. There's no data. So I'm just wondering if that would be a useful thing. Another thing is, because I'm trying to help you guys with the model master file idea here. Recently, a colleague of mine, Polli Corcels, took one of our models in our platform at Nova Discovery, and she translated it into MATLAB. We wanted to do some benchmarking and other things. She did me the favor of creating it in a live script, which I think many of you know. It's just basically you get the script, you punch the button, and it runs everything, and it documents everything. I'm suggesting that, and I talked a little bit with Dr. Hooker about this morning, so if it's a good idea, it's his idea. If it's a bad idea, you can blame me. The idea would be to have a model master file that's disease progression, for example, and then have a drug model in that model master file that would be either something that's published, it's out of patent, it's not controversial, or it's well-known, or the sponsor's published a model. The point would be that the model master file wouldn't just contain the model structure and some parameters and parametric distributions, but it would also include a script, data, preset graphs. It would draw the people that would be using the model master file through the process that the FDA needs to see to make an evaluation. So 2 questions. One, would it be useful to have disease progression QSP models? The second, that are industry-wide, and the other is, would that live script idea be useful in the model master file context?
Yeah, I like your idea. I think there's a lot of potential for the model reusability for QSP modeling or those disease progression modeling. I will refer this Hao again.
Yeah, I just want to quickly touch base on the second question. Maybe some other, like CG others will chime in for the first question about the industry standard. The second question you're talking about, can we actually get acceptance for disease models with the codes and everything that's been public and you can use it? That's exactly what the Fit for Purpose program is, where we have the program being set up that way. The first program is under Alzheimer's disease model. That is exactly what you said. And it's being run by CPath, Critical Path Institute. And we, at least that's what the intent is, to release everything in public. So the codes and everything, data sets, I don't know how you get that access to them. Maybe you need some types of registration to them, but that the intent is to release that to public and everybody can use it and the codes are available and everything's available. That's my understanding from what they told us. Yeah.
Yeah, thank you, Jim. And you know that I've been a passionate fan of public models, but also recognize that there's also proprietary models that have great value too. So I do like the idea of coming to an industry pre-competitive agreement about models that we might use. It doesn't have to just be industry, it could be regulatory and also patient groups, advocacy groups as well. I think it's a community effort in that. We do have an example where we submitted a QSP model of the immune response to COVID-19 to the agency as part of our EUA submission. Maybe Jiang can comment on what it was like for them to review it. However, what we did do is we had several publications, peer-reviewed publications, before we made that submission, so that we were hoping that that would allow the agency time to see what might be coming with our submission. And also, with our submission, that did include the proprietary drug information, which was not in our publication. So we've kind of worked a way of how we can do this internally with our internally developed QSP models and interacting with the agency. So.
Thank you, CJ. Actually, we got your QSP first from the GitHubs, right? I think Joga has something to add.
Yeah, I just wanted to add a proposal for the agency to consider, which is that while, let's say you embark on this MMF path, it's going to take time for the sponsors to kind of digest and then prepare themselves to do that. So my proposal is, OGD, for example, gives $17 million worth research grants to different organizations. Make it a requirement. You're going to get federal research money, submit the files in this requirement, according to MMF. You get experience through that. Then we'll be in a better position to come up with these standards.
Can I make a last comment?
Sure.
I just want to share, yesterday, Marie had shared in the table that, for long-acting injectables, he developed some models, and then the interval to develop the model is about 2 to 3 years, loss of communication with FDA. The model could be generic, and after this initial effort, say if another applicant wants to use the same design, a model integrated design to support another product, I think the cost of saving could be, I'm the expert already, negotiated with FDA for the past 2 to 3 years. Now things coming to a stage that can be widely implemented. I think I do see a very big cost of saving, even for QSP models. Initially, it's very complex with hundreds of equations. Then effort being taken to evaluate that, to validate that. So if there's a master file, that can certainly give lots of cost saving in future, even though, Hao and Jiang not totally agree with the model, but at least there is some regulatory discussion of which portion of the model can be recycled and what data is still needed to substantiate for the regulatory use. I think that will, even with that will cut lots of time.
Thank you very much. I guess that's the first step, I guess, is to initiate the discussion. I think we still have a lot of questions, but we can continue that offline. So I'd like to thank you, my panel, and the great presenters for this their contributions. Thank you very much.